So cutting the DNA into fragments with restriction endonuclease enzyme, right? So we talk about that step a little bit. So now we don't we keeping the DNA long fragment is not serving the purpose, because your insulin gene could be anywhere, could be in here with that fragment, right? In somewhere else with other fragment, right? It's a small piece of DNA compared to the whole DNA molecules. So it's in here. Some of them, right? Some of the chromosomes, most of the chromosomes, that gene is not there, but their DNA fragment present in here as well, because we didn't select one by one, right? We just all together extracted. So we call them genomic DNA. Genomic DNA means whole cell DNA, DNA of the whole cell. What is the word? Genomic DNA. So when you say genomic DNA, it will come as a large fragment like this, very large fragment, right? So, but we have to make sure this DNA, genomic DNA, is in good quality. So, what do you mean by good quality? Because if the DNA, right, if you don't handle it properly, for example, if you don't put in ice, right, or if you handle carelessly, probably this DNA would be fragmented. We call shared DNA. So it is already, right, especially when you extracting a tissue, it already have endonucleases, right? Restriction enzymes already there. So what do you have to do the first place? You have to inactivate this. There's one step. That step is for inactivate all existing endonucleases. Otherwise, your DNA would be very, very, very bad quality, right? It is cut from everywhere. So then no point. So once you've done the procedure rightly, so you got to check it. So if you want, you can add these steps as well. So look at this diagram I show you. See, this is a marker. The small fragments running in front, and the larger fragment running behind. So, right? So the DNA, there is a DNA well. See, we're loading into these wells, and DNA is running from negative electrode to the positive electrode. Right? This is a gel. This is a negative electrode in this side, positive electrode in this side. DNA running from negative direction to positive direction, because DNA itself negative. DNA itself negative charge. DNA is negatively charged. So good. So if it is not fragmented, the all of them together like a big chunk, big bulb, big bulk like this. All right. Probably this is not human DNA, or this is not mammalian DNA either. This is, I think, bacterial DNA. But if your mammalian DNA size is big, if size is big, the fragment is running very slowly. Maybe the bulk DNA coming here. It is human DNA. Anyway, you got to test this quality of the DNA. Checking the quality of the DNA is important, right? So these are additional steps. If you want, you can write. So then, in case you lose the marks for other steps, so you can gain the marks for these steps. Right? If you understand, you can actually add up a lot of points in there. Got it? So the next one is Digesting by restriction enzyme, as you saw the list of the items. So digesting it by restriction enzyme. So, okay, this is our DNA, and most of the DNA, our fragment is not there. Our fragment is only one chromosome, right? So one chromosome has two copies, homologous chromosome. So out of 46 chromosomes, only two chromosomes having a copy of our insulin gene. Sometimes important genes like this has more than one copy, but I think insulin gene unfortunately has one copy, right? One copy somewhere. Only one chromosome, homologous two chromosomes have that copy. So now this is the gene, right? But we need to take the gene, cut out of the gene, intact or entire, right? So entire gene has to cut out, or intact gene has to cut out. So we have to select a restriction enzyme, cutting closer to this end, and also cutting closer to other end. Not cutting inside. All right. How do you find that? Well, you can do from the database. So you just input the DNA sequence of your gene. So you can copy it down from the database, and then you select the selected enzyme, number of selected enzyme. Just select EcoR5, for example, enzyme EcoR5. Okay, so your gene is from here to here, but you actually enter or input limit either side region. We call flanking region, right? Sort of word, flanking region. In your words, you can say the DNA sequence either side, right? Flanking DNA, or you can say adjoining DNA. No problem. You can say adjoining DNA. No problem at all. So adjoining DNA sequence together we put. So then the program will give you a map. The program is this is your gene, right? So this place is a restriction site. None of here. Then another one here has a site enzyme EcoR5. Very nice diagram they give. And then if you put another enzyme in 3, okay, check that too. We ask the program to do that. So then they will get a different color for that. So the in 3, one site here, one site here, and another site here. I just put H for that in 3. Likewise, you can check easily. You can check large number of restriction enzymes. Then you say, oh, no good. See, it's cutting your gene. EcoR5 is better. See, it's sparing your gene. Right? So let's cut from EcoR5. And we cut the restriction enzyme EcoR5, EcoR5, cutting your whole DNA, complete DNA. So your complete DNA will cut into small pieces. All right. But one thing of sure, it don't cut your gene. You are sure of that, right? So we go back and see what is the next step. I think agarose dual electrophoresis. Right? Go and have a look. Cutting DNA, separation DNA fragment from gel electrophoresis. All right. So material called agarose. Agarose gel electrophoresis they're talking about. I got diagrams this time, so I'll show you what do they mean by. So this is what you actually getting from. So this is a marker. That marker, every single band is documented. Right? The manufacturer of this marker will tell you about every single. So two here, two here, two here, two here, two here. Likewise, they will give you the size of all the single fragments, and then compared with that, right? Because this agarose gel separate the DNA fragments by the size only, meaning that bigger fragment running slow, smaller fragments running faster. So slower the fragment, which is from negative electrode to positive electrode. So the smaller fragment very quickly coming to the edge. The bigger fragments coming very late. So then you get something like this, right? So actually the genomic DNA card, this is not genomic DNA, probably this is actually plasmid DNA card, right? Plasmid DNA is small DNA. But genomic DNA is a big DNA. So then you get every single well loaded with restricted fragments of genomic DNA of human being, right? And it is actually large number of fragments from here to here. Whole the pathway full of fragments. All right. So, but it's similar to this. But by the time, so extraction of human DNA, how do you get the sample? Huh? Can I ask that question? So how do you get a sample sample of tissue to extract DNA? Do you know that about it? If you want to extract human DNA, so what is the best sample you can get? Yeah. Hello. You there? Yeah. That is human blood. In blood, although red blood cells don't have nucleus, you know that red blood cells don't have nucleus. If there is no nucleus, there is no DNA. Only white blood cells have nucleus. There's a small technique, very small technique to separate white blood cells from red blood cells, right? So because once you put into water, all the red blood cells lyse. White blood cells don't lyse. So they once you centrifuge, all the lyse red blood cells go to bottom because they have hemoglobin. Hemoglobin is very heavy. But white blood cells, like a small layer depositing on top of the red blood cells. So you can, by pipette, you can carefully take only the white blood cells, right? So then white blood cells can use to extract DNA.